Dyslexia is a wonderful example of an important medical problem that spans both neurology and psychiatry. For us at UCSF, that is a goal, to break down these artificial silos and bring groups of smart, motivated, and collegial people together.
This combination of very sophisticated psychiatry, biological psychiatry, thinking about genes, chemicals, in combination with a very anatomic-oriented and language-based group in neurology is unique. This isn't happening anywhere else in the world.
The integration happens in very many ways. So it can happen in the exam room with a pediatric neurologist and a child psychologist and a child psychiatrist all working together in evaluating a child and coming up with a treatment plan together. It also happens every day in laboratories where you'll have a molecular biologist talking to a computational neuroscientist and then have a psychiatrist right there.
Our mission at the UCSF Dyslexia Center is to understand better, to uncover the devastating pieces of dyslexia so that we can target each of those pieces. So much of research is something that takes place in the laboratory, but it takes a long time for that information to get translated into an actual setting where it's useful.
As a neurologist and as a mother of a dyslexic child, I'm really excited about what we can do here, the novelty of the work we do, combining the technology, the brain science, the imaging, the biology, to really understand the strengths and weaknesses of dyslexia and bring this back to empower families and schools and children.
When we're looking at a child with dyslexia, reading problems don't happen in isolation. It comes with other things. For example, they might have memory issues, learning issues. They might also have attentional issues. What we're trying to do at the Dyslexia Center is to identify their exact weaknesses, but also their relative strengths, so we can come up with this optimal and comprehensive but efficient way to instruct and intervene each child.
One of the things about dyslexia is that it's a family journey. It's not just about the student, but it impacts everybody in the family. My son was diagnosed as a dyslexic learner when he was 6 years old, and he was lucky enough to have attended the Charles Armstrong School, which is a school that specializes in teaching kids who are dyslexic or have other language-based learning issues. I was president of the board of trustees, and I started looking around to try to figure out how could we improve our curriculum. I approached UCSF and I said, you know, what's being done out there is not good enough. We need a research center that's driven by the needs of a school so that the outcomes will be very practical. And we need to get those outcomes to happen quickly because we have a crisis.
Our current flagship project is called the Phenotyping project, and what we're essentially trying to do is take a number of dyslexic children and then studying them in real depth, from the genetics to neuroimaging to cognitive and behavioral, environmental, and social emotional factors.
Phenotyping refers to the way that we can understand the outward expression of a disorder. We can see the different aspects and piece them out so that we can target them. In a sense, we're subtyping.
When we put that together with the Armstrong School, then we're able to look at outcomes, what happens when we intervene, how to make those interventions better, and we can also work to bring the technology that usually would be isolated to the academic setting and take it to the classroom at the Armstrong School.
One of the things that has the biggest potential for impact on the dyslexic learner is the likelihood that we'll be able to detect and diagnose dyslexia at a much earlier stage, before the symptoms really start.
People talk about it all the time, how you're going to go from bench to bedside. That's what everyone aspires to. There are very few places where it's really happening on a moment-by-moment basis. And part of that is the culture at UCSF, and part of it is the environment that's been built here. And I think in the long run, it's going to be really the key to addressing problems that have been really almost intractable for decades.
Hi, I'm Adi. Nice to meet you. Have a seat here.
Our lab is conducting the speech feedback study here at the Dyslexia Center, where we're examining how hearing yourself speak affects how you speak. In the study, we carefully control the audio feedback that subjects hear while they're speaking. We pick up their speech from a microphone, and then we pass the audio feedback back to them on the headphones that they're wearing. And then we record the speaking that they're producing. The reason we're doing this is because there is this idea that dyslexia can be associated with auditory processing abnormalities. The point of view of a speech researcher suggests that dyslexics may differ from controls in how they respond in their speaking to hearing themselves speak.
How was it?
The hope is that this will provide us with clues that will gain us a better understanding of the precise mechanisms in dyslexia.
In dyslexia, we think that there's probably a range of emotional strengths and weaknesses across different individuals. So some people might be really attuned to social cues, whereas other people might have more difficulty understanding complex social environments. We're really interested in how the body responds physiologically during emotion, so we measure things like heart rate, respiration. One of the measures that we use is an eye tracking device, and we're interested in seeing where people are looking in the visual field to see what parts of an emotional scene or emotional cue that they're taking in.
So the glasses actually measure pupil size, and when you react to certain things, they either dilate or constrict. And we also test what they're actually looking at on the screen.
By better understanding who is strong in the area of emotion and social understanding and who might need more help in this area, we can both understand the biology of that individual person's strengths and weaknesses and also tailor interventions to better help that person in school and in life.
For the Dyslexia Project, we have a battery of measures of spatial cognition that measure a variety of different components of spatial processing. How we understand what something looks like and where it's located. How we can remember those things briefly in our mind and over longer periods of time as well. We're interested in seeing how dyslexia might affect spatial learning, and in fact may even enhance performance on some of these tests. Most of our measures of spatial cognition are computer-based. So some of those are on tablet computers, and others look like driving video games. We want to understand for these individuals, where do they have strengths? Where are they really no different than people without dyslexia, and where are there, if any, weaknesses? And this information can help us understand the different phenotypes of dyslexia. And this is an important step both for treatment and for helping with compensatory mechanisms.
In the last decade, we've learned from the MRI that dyslexia really comes from the brain, and technology now is such a high level that we can look at really high resolution of how the brain functions and how it looks, and we have great people, engineers, who spend their life using this technology. The resolution is so high that we can look at millimeters in the brain, and we weren't able to do that before. We have the computer power to analyze thousands of images all together, to look at connections between brain areas that we weren't able to look at before. So I think it's a really exciting time. It's not just to look at the difficulties of the dyslexic brain, but we can really help kids and adults look at their strengths. We can look at the neural mechanism of how they do things differently at the individual level. As a neurologist and a cognitive scientist, I think it's just really exciting that we finally have the technology to do what we need to do to understand dyslexia better.
The brain is a very specialized organ. It means that each millimeter is responsible for a different function. Post-mortem examination is the exam that provides us the best resolution of what we could see in the brain. Most labs working with post-mortem tissue, they take a brain and they make small pieces of the brain and then they try to put all together. We developed a protocol in which we can process the brain without having to make small pieces. Therefore, instead of having to glue all the pieces together, that is prone to errors, we can see the whole brain in one picture. These results are much more reliable, easier to compare to controls, and even better is the best way to compare images that we acquire in vivo using MRI, for instance, with exactly what is going on in the brain. It's so important to have post-mortem examination in the study of dyslexia together with the other exams, because putting all of them together will give us the best understanding of what is causing the symptoms of dyslexia.
The case conference really serves two very important functions for the dyslexia program. One is for the families, for the children who participate and their parents. It's our chance to share with them what we've learned about each participant. What we do is we sit down with the family, we go through the brain imaging, we go through the testing that we've done, we explain what it all means and how it fits together within the context of what we've learned about dyslexia so far, and answer their questions about how best to help in school. The other important function of the case conference from the researcher's perspective is that it gives us an opportunity to talk to each other and sit together and see what we've learned about each participant that comes through the program.
So he doesn't seem to have like the oppositional type of behaviors.
For the phenotyping project, the case conference is a really vital part because it's our opportunity as researchers to sit together and to discuss what we think is the phenotype of each particular case. I think we've come to this project not with a lot of preconceived notions about what we think dyslexia is. I think we let the cases inform us as to what it might be, and we've used that experience over the course of the project to build our understanding of what the various types of dyslexia might look like.